Hello, everyone. Good morning, afternoon, or evening to everyone. Welcome to our latest Novogene webinar. And thanks for joining. My name is Adrian Cordes. I'm an account manager at Novogene in Germany. And it's a very big pleasure to co-host this exciting webinar today with our invited speaker, Gianni. Very exciting. Big thanks for being here. Looking forward to this webinar. And he will present today some insights on his research, which is about metagenomic sequencing illuminates gut microbial contributions to colorectal cancer. So before we start, let me give you some general infos about this webinar. And a little bit of introduction to our guest today, and also a little bit introduction to Novogene. For those who haven't been a while with us, we host quite a series of webinars, and you can find them all online on our website, free to watch. You can check it out. So the main presentation from Gianni will run around 40 minutes today, and during the talk, you can feel free to submit questions in this Q&A feature, which is located on the bottom of your screen. So you can upvote the questions by clicking the thumbs up symbol next to it. So if we don't have the time to answer all questions today, we will make sure to follow up on them, and we will answer them on a follow-up email afterwards. So feel free to question, of course. Please don't use the hand raise function for any questions, or if you encounter technical issues, please reach out through the Q&A section and we will message you directly. And we will also share the slides and the recording of the webinar via a follow-up email later this week. And you can also get a certificate of attendance. We will share a link you can click and you can directly print it out. Finally, there will be a short survey at the end, which will pop up after this webinar. So we would appreciate if you take a few minutes to fill that out or feedback us. That will help us to improve the content for future webinars. But yeah, now let us getting started with a short introduction to our today's guest, Gianni, who's from Greece. He did his PhD in Greece in 2005, and he had several, let's say, locations he have already been, starting in the Technical Uni in Denmark, where he did his postdoc, and also associate professorships in the Center of Molecular Biotechnology, or the Center for Molecular Sequence Analysis and Novo Nordisk Foundation Center for Biosustainability. He has a professorship in the University of Hong Kong and the Friedrich Schiller University in Jena. And actually in Jena, he is now leading, and he is the head of the group department Microbiome Dynamics at the HKI, the Leibniz Institute for Natural Product Research and Infection Biology, or also called Hans Knoll Institute in Jena. He's also editor-in-chief for the Computational and Structural Biotech Journal. For his main research, he's very interested in microbiome in health and disease, in systems biology of infectious diseases, also in personalized nutrition. And to quote him, he is very eager to understand the role of gut microbiome in human disease and engineering the gut microbiome for therapeutic purposes. He is very connected in Jena, but also worldwide. So also very open for collaborations if you want to connect with him. One slide about our, let's say, webinar series, Genomic Research Live. We hope to make it more often. Novogene wants to, let's say, make connections between researchers happen by co-hosting this webinar and serving as a kind of a platform. We want to give you as researchers this platform to expose your research on a globally level with hosting these live webinars and expert Q&A sessions. We want to discover by this and discover and exchange innovations and make collaborations across borders happen. Yeah, feel free to follow us on that. As a very, very small introduction to Novogene, Novogene is one of the leading companies providing NGS solutions on a global level. So we have laboratories 10 laboratories actually worldwide, spanning from the US, Europe, and Asia, in over 90 countries on the 6 continents. Just to mention, for our Europe locations, we have a lab in Cambridge and very new, starting or like opening the beginning of last year, we have a brand new lab in Munich in Germany. The service portfolio of Novogene spans basically the whole spectrum of NGS, basically, starting, of course, from all the human-related genomics solutions like whole genome, whole exome, the whole world of transcriptome, from mRNA to non-coding over isosequencing, and very recently also spatial transcriptomics, single-cell RNA or single-nuclei RNA. We can help you also with the whole microbial world of services, from whole genome or metagenomic sequencing, which is the topic for today. We have a lot of services for the epigenome in-house, like enzymatic methylation sequencing, whole genome bisulfite, etc. Of course, we can sequence all species possible, animal, plant, everything. And very recently, we added proteomics also in our portfolio with the Olink technology. And Novogene, of course, is always expanding the portfolio in the more omics direction. A few points I want to mention why to choose Novogene. Of course, what is very important to us is the quality of the results. Of course, we will make everything happen to create reliable results. For example, having all the quality measurements from Illumina and the other technologies in place. And Novogene has a very, very large capacity of sequencing. As you saw, the labs we have worldwide, and we are always expanding to make the capacity happening with our machines. So there's a very high automation system also in our pipelines to have this throughput. So no matter how big the project is, we can do it, and we can do it also very fast. I will come to that also. Latest technology is very important to mention. Normally we have now Illumina, PacBio, and Oxford Nanopore technology with the state-of-the-art sequencing machines, which is Illumina NovaSeq X, PacBio Revio, and Oxford Prometheon. But also there, Illumina, Novogene, of course, is investing in the latest technology on the market for you to know. So it's not restricted, of course, and the world of NGS is moving very fast also in new technologies. As I mentioned, we are really, really fast due to high automation system and this high capacity we have in our labs, which is normally faster than local cores or a big majority of service providers. And we have a big team in-house of bioinformaticians, so we are also happy to help you with some expert analysis there. And one last slide to mention here on a global level, of course, but for Europe, we have a lot of contact person, like a local representative, which are very, very happy to help you, so that we understand the need of really close discussions of your NGS project, which is really important because it's not sometimes very easy to find the best solution. But we are there, and feel free to contact us, and then we can also come to your office. Great. Thanks for listening to the intro, but now stay tuned. I will hand over to Gianni. Feel free to share and start your presentation.
Okay, yeah. All right, so you can see the full screen? Everything is fine? Yeah.
Yes.
Yes, all right, okay. Let me also remove this from the top. Yeah, so hello also from my side. I'm really happy for the invitation from Novogene for joining this very known series of webinars, and also Adrian for the introduction. As Adrian mentioned, I'm going to talk about the microbiome and colorectal cancer in this presentation, and I hope that it will spark nice discussion afterwards. That's always the most exciting part when people start doubting about your findings. Uh, so yeah, humans we have always been very concerned about our health, and we are always trying to figure out which organ of our body is the most important in order to keep us healthy. And it's not a recent thing. It's actually from a very old times that we realized that the gut is an extremely, the intestine are an extremely important organ for our health. So if you think the ancient Egyptians, how they were treating the famous dead people of their culture, it's pretty interesting. When someone very important person dies in ancient Egypt, then they will wrap that person with linen, but they will remove first the intestines. They will remove the intestines and they will put them in a special jar, because they consider this as extremely important also for afterlife, that your intestines are intact. And they even have actually a god that is going to protect your intestines in this jar, so you can use it after death. Now, the ancient Egyptians are not the only ones that believed on the importance of intestines. Also, the ancient Greeks. I mean, as a Greek myself, of course, I would have added something for the famous Greek doctor Hippocrates. Hippocrates have said many famous things, but one of those is that all diseases actually begin in the gut. That was a very strong statement, systems level thinking of human diseases. And this is not the only thing that Hippocrates said. He also said that your diet can work as a medicine as well. And if you think these two are very much connected, right? If your intestine is the center for every disease, then the diet that it is ending up in your intestine can also be the medicine. So it was very smart thinking from Hippocrates. And so now here where we are now, it's interesting that we say that the microbiome is a relatively young field. We mean it's around 2,000 years, right? That's how long we believe that the gut is important. But there is big difference. The big difference is that with the next generation sequencing, we started understanding really what is going on in the gut. We figure out that actually we are not alone, and especially in the gut, we have thousands of strains that are living, bacteria, fungi. And these are the mediators of what Hippocrates was saying, that the intestine is the start of many diseases. And we use the microbiome for different purposes now. So we can use the microbiome as a diagnostic. You can classify people. Whether they are healthy or not by looking just their microbiome. And for some diseases, this is very important, because there are diseases like one disease that I'm working, that it is non-alcoholic fatty liver disease, that it's very invasive to diagnose the stage of the disease. So if this was possible by just looking into your gut bacteria with a stool sample, it's extremely important. You can use the microbiome for stratification of patients before treatment, especially for immunotherapy, for cancer immunotherapy. We know now that based on your microbiome, we can predict pretty accurately whether you are going to respond or not to immunotherapy. And this is also very important because time is very critical for cancer patients. So you don't want to waste 6-8 months with a therapy that you know is not going to work. So that's also very, very important. And also for monitoring the response to treatment. So you can follow whether with some specific microbiome biomarkers, whether the treatment is really efficient for that person or not. So these are the main areas that the microbiome has gained a lot of attention, and this is more or less what we also do in my group. So we try to contribute to this new field that we call it ecosystems medicine. I don't have in my lab a favorite disease. So we work with metabolic diseases, we work with cancer, we work with infections, but we do have a favorite research approach, let's put it like that. So this cycle that you see here is the classical approach that we have in our projects. We will more or less almost start with humans, with some kind of clinical study, and then we will use Novogene very, very often, as Adrian said, that they have really a fantastic portfolio of omics to generate as many microbiomic data as possible. And then the next thing that we are going to do, my lab is very strong, I would say, computationally. We'll try to make sense from all this data. We try to understand exactly the mechanisms, how from this omic data, how the bacteria can really affect the host. So it's both high throughput analysis of the host profile and also of the microbiome. And I would say that this is the work that we have been doing the last 5-6 years, but in reality, I should update this figure because now we have entered the phase where the circle continues, because we are very much interested on how we engineer the microbiome. How can we shape the microbiome to bring it to a direction that it will be beneficial for the host? From all the different omics that we measure, I think one of the most fascinating, for me at least, is the metabolomics, is trying to understand all this, or characterize, I would say first, this huge metabolic output of the bacteria that they live in our gut. You can imagine that we have constantly in our gut a reactor that is working, is getting the diet and is getting other xenobiotics and is producing different molecules all the time. And these molecules, the majority of those belong to the secondary metabolism. So they have structures that they may look like a drug or they may act like drugs. And there was this recent paper in Briefings in Bioinformatics that I have put here for the ones that are interested. So they tried to make a summary. What do we know? How many metabolites we have characterized that are really produced by gut bacteria? And it's, I think, I don't remember exactly the number, but it's more than 3,500 metabolites that are constantly producing. So it's really important to put attention on this. Sorry. Apologies, there was a technical thing. So that's one of the metabolites of the omics that we pay a lot of attention. That we'll talk more today about it. So the project that I'm going to talk about today is a PhD project from a very bright student, Lin Shu, and it's exactly on this subject: how gut microbes and their metabolites can affect colorectal cancer. We know already a lot about gut bacteria and colorectal cancer, so this is not the first time that someone decides to look into that. We know already, I think it was in 2019, where Nicolas Segata lab published in Nature Medicine a paper where they looked into colorectal cancer patients and controls, and they show that if you do metagenomic sequencing, you can develop a machine learning model that can predict whether you do have or not colorectal cancer. So this is on the diagnostic part. Then we know that there are specific gut bacteria that they can trigger CRC development with different mechanisms. We know that E. coli, for example, can produce a toxin. Specific E. coli can produce a toxin that can damage your DNA. We know that Fusobacterium nucleatum also can induce CRC. So this goes to the second part of more or less mechanisms and potential therapeutic approaches. And also we know that there are gut bacteria that can influence immunotherapy. It's also very interesting that there was a paper recently that indicated that actually Fusobacterium, even though it is involved in CRC development, its presence can influence positively therapy response. So things are not really clear yet of how all these mechanisms work. So why did we start this project? I mean, there are of course a lot of different things that have not been answered. This is just the seed questions that we had when we started, but we learned many more during the project. So most of the projects in metagenomics are based on stool. For obvious reasons, it's very easy to get it. So that's why. But we wanted really to understand how reliable is to look at the stool of colorectal cancer patients for understanding mechanisms of cancer progression. The second question that we had and we wanted to answer is why some bacteria like really to sit and colonize the tumor tissue. And the third one is, can we identify new mechanisms that gut bacteria can promote colorectal cancer? So that was how we designed the study. And since we wanted to do a very deeply phenotype cohort, we didn't go to very large numbers of patients. So we worked with a cohort of 77 patients. Very nice distribution of different things. What I mean is that they cover a broad range of different ages, males and females, all the four cancer stages, both the tumor site, both in colon and rectum. And then we really took advantage of technological developments, and we said, all right, let's characterize as much as we can this cohort. The beauty was that besides having stool, we had also tissue. So for every patient, we had the biopsy from the tumor and also from the normal mucosa. And we did shotgun metagenomics for that. We had the stool. We did metagenomic sequencing also from the stool for the bacteria, but we also did ITS sequencing for the fungi. And then from the serum of the patients, we did a very detailed metabolomic analysis with more than 2,000 metabolites. We did separate lipidomic analysis, almost around 2,000 lipids, and an okay characterization of the immune system with just 40 cytokines. And we had additional information for these patients from their metadata. I started with the fungi, not because the results were that amazing, but it's anyway, fungi have been studied much less. So already is a little bit, I think, interesting to see what could be the contribution of fungi. So you have the 4 stages of cancer, and the analysis in the stool was based to see which bacteria or fungi are involved in the progression. So the comparisons that we do is the abundance of species from stage 1 to stage 2, from stage 2 to stage 3, and also ordinal regression. Are there any particular species that you see them increasing during the progression of cancer or decreasing? As you see, the fungi didn't give us a very, as I said, spectacular result. There are a couple of fungi that we see a trend in the ordinal regression, but most of the changes that we could observe with confidence in the significance were really from stage 1, from the most mild stage to stage 2. We found 4 species that were increasing their abundance. And when we look into how these correlate with cytokines, we found that all these 4 species were correlating positively with TIM3. And for the ones that are not very familiar with these cytokines, TIM3 is a negative regulator of anti-tumor immunity. So it makes sense. Species that increase in the more severe stages of the disease also correlate with cytokines that are not helping you when you have cancer. Then we did the same for bacteria. Of course, for bacteria with metagenomic sequencing, you have a much larger set of species to work. Actually, we are talking here about subspecies because the sequencing was very deep, so we could assemble MAGs, metagenomic genomes, and look how they change again. Similar analysis as with the fungi. And here you get much more. You get both unique gut bacteria signatures. What I mean unique is that species that they only change their abundance from stage 1 to 2, or from stage 2 to 3, or from stage 3 to 4. But you also get species that they have a more consistent pattern, that you see them increasing always when you go to more severe cases. But the majority, again, were pretty unique. So most of the species, you would see them only in one, changing from one stage to the next one, which I personally found very interesting. And I couldn't believe it that is happening. So what we did was the following. We discussed and we thought that what is probably happening is that even though you have different species, that characterize the progression from one stage to another, in reality, these species have the same functional potential. It's just different species, but they can do the same. So since we had the MAGs, we did functional annotation of all the subspecies, and we said, okay, now let's cluster based on their functional potential. And what you get at the end is this heat map. I don't remember if this is a complete heat map or just a part of the heat map, but you see the last column at the right, which is either blue or red. Blue are species that decrease while the cancer is progressing. Sorry. Red is species that they increase their abundance with the severity of the disease. And you see that they are clustered beautifully, right? So, and you can see also the functional annotation of the pathways that are enriched in one or the other. So it's our suspicion goes through different species, but same at the end. Then we looked also the systemic impact that gut bacteria can have on all these metabolites and lipids that are circulating in your body. And of course, they are playing a major role on how you respond when you have cancer. So we took the functional profile of the gut bacteria, and we try to see whether it correlates with the profile that we have of the metabolome and lipidome. And indeed, you can see that there is a strong correlation and a positive correlation, confirming that what happens in the gut really doesn't stay in the gut, right? It has a systemic impact in your whole body, the changes that are going to happen in your gut bacteria. I see that the chat is it something that I should stop or I continue? Because I see my things popping up. You can still hear me and everything is good?
It's all good. We can see it.
All right. Okay. Because I see constantly the bar coming, notifying me. All right. Then I continue. And another thing that we did is that we looked into the metabolites and lipids, and we look at them then in a very interesting way. We built it something that is called differential coabundance network. So instead of looking of how each metabolite or lipid is changing while the cancer is progressing. We look at it as a structure. So you create, if you think about it, you create a network of metabolites and lipids in stage 1. You do the same in stage 2. And there is a mathematical way that you can compare these networks, and then you can see what is differently regulated when you're moving from one stage to another. And then if you try to investigate further these networks, we could see modules that are common in all the cases when you are progressing from one stage to another. And I think a very interesting module was this valine, leucine, and dissolution biosynthesis, that we found that it is always a structural change that is happening in your metabolomic profile while the cancer is progressing. And then if we go back to the functional profile of the gut bacteria in these clusters that I show you before that they are forming, we could see a strong correlation between changes that are happening in the microbiome, in the functional potential of the microbiome, with changes that are happening in the metabolite, and especially this valine, leucine, and dissolution. And you will see that again later on why we are so much interested about this. So that was the stool. That's again, I think many people have looked into the stool. I wouldn't say that what we did was the most novel thing, but I think the way that we approached it already gave us a lot of information. But I think the most interesting part came when we did the shotgun metagenomic of the tissue, because most people have been unable technically to do it. Not that they didn't want to do it, but they ended up with 16S, because when you try to do the tissue microbiome, you have huge amount of host DNA. So we needed to really optimize the pipeline to do that. We needed to go extremely deep in sequencing. But even when we did all this, we were wondering, are we really sequencing what is there? Or we are sequencing things that are basically the dirty hands of people that they were handling the tissues or the pathologist or all the things. So the first sanity check that we did was to compare the microbiome that we got, the composition from the stool, and the microbiome from the tissue. Because, of course, even though you don't expect to be the same, you expect that these two are correlated. They are sharing the same space. And indeed, there is a strong correlation and statistically significant when you compare the stool metagenomics with the tumor metagenomics, the stool metagenomics with the mucosa metagenomics, but also the tumor with the mucosa. You will say, yes, why it should have been different? I agree with you. But I think it was important that it was the same, that we saw this and made us more confident to continue working with this data. But at the same time, when you compare the microbiome of the tumor and of the healthy mucosa with a beta diversity analysis, is significantly different. So that's now that you get even more excited. So different microbes are sitting on the tumor, different microbes are sitting in the normal mucosa of people. And then we started looking at the species level. What are the changes that we see? Now, if we take the tissue for a start, as I said, for every patient, you have the tumor and you have the normal mucosa. So you have a perfect control where you can look for significantly different in abundance species. And you will see at this kind of heat map on the left that there are some bacteria that it doesn't matter what stage of cancer you are looking at, you will find them either increased or decreased. Take, for example, the top two. So I don't have the species names because it's unpublished data. Of course, we are very close to submit this work, but it's fine to say to you that these Fusobacterium are the two famous Fusobacterium nucleatum and necrophorum. So this work as reference also for us. So you can see that it doesn't matter in what stage of cancer you are, these bad species are, you will find them significantly different in abundance. Now, the interesting thing is that if you look at the stool, now some of you, the ones that you were paying much attention from the very beginning, you may say, but you didn't have any healthy subjects. How did you compare the stool? That's true. So we matched 1 to one of our patients with publicly available data to match them in terms of age, BMI, gender, ethnicity, and we compare the metagenomic data in the microbiome profile of our patients with healthy patients, the stool. And it's interesting that for many of those species, you get exactly the same information if you look at the stool. So you see for the 2 Fusobacterium, you will find them even in the stool significantly higher. Now, the interesting part comes on the heat map on the right. And again, these are just snapshots. There are many more bacteria that are significantly different. And then you can see that you have bacteria that are either constantly red or blue when you look at the tissue, but nothing comes significant if you look at the stool. Not only that, if you look at the very last column, I don't know if you can see my mouse, this one that says stool abundance, the majority you cannot even detect them using the stool. That's, I think, very, very interesting. And for me, it was also very interesting that many of the species that you see on the right are actually oral bacteria. So what we did afterwards, we said, let's take species, let's make a selection of species from here that we have available in the lab and study them more. So we selected 25 species that we had available that were differentially abundant in our tissue analysis. Half of them approximately were oral bacteria, the other half were normal gut bacteria. We grow them individually in vitro, and we did untargeted metabolomics to see what is the metabolic output. And you see that they cluster differently. So other the metabolic properties of oral bacteria that you find in the tissue, other the metabolic properties of the gut bacteria that you find in the tissue. I think that was already pretty interesting. But if you try to put them all together and see how they cluster, you see again this very fascinating pattern. So you see the last column again, I think for me is always the most interesting. Red species are the bad species, blue species are the good species. So there's the species that are depleted from the now we are talking from the healthy, right? So not for the progression, it's tumor versus normal mucosa. So the good species are clustered together, the bad species are also clustered together. And you have this interesting, very last group that both red and blue species are there. So we decided, okay, this is the one that we want to look even more carefully what is happening. Now, before I go to the last set of species, we looked into a functional enrichment analysis based on the metabolomic data for these different clusters, and then you start seeing the valine leucine and the isoleucine to be in several of these groups, and you also see some other pathways that, again, I'm sorry that I cannot tell you all the truth in this presentation, but because it's unpublished, but you see some other pathways that were very interesting to be enriched. So then we said, all right, we need to prove what is happening. We have to go to the mice and see in a cancer model. We use the MC38 tumor line to see if these species that we believe that they promote CRC, these oral bacteria can indeed in a mouse model do that. So the way that you do it is that you inject the bacteria in the tumor directly in 3 different time points, and you look into what happens to the tumor. So we injected 4 oral bacteria, and only 1 is working. That's very interesting. So even though you see them enriched in the tumor, of course, it doesn't mean that they are promoting the cancer. It could be just sitting there. And this is another thing that we also explored, but I will not use it for this presentation, is why these species are there. But from the 4 species, only Leptotrichia, a species of Leptotrichia, was inducing the tumor. And then the next thing is how Leptotrichia does it. So the obvious first experiment that you do is you try to understand, is it something that it is produced or consumed by leptotrichia in the tumor and is inducing the cancer, or is it something that is in the membrane, so even with a contact is enough to induce the tumor? And so the experiment that you do is that you repeat it, but you also add heat-killed leptotrichia to see if also the dead species is also promoting, or the medium that it is. So instead of giving the whole bacteria, you grow the bacteria, you take the condition medium, as we call the original medium, the supernatant, and you inject the supernatant. Because if it is something that it is simply produced by leptotrichia, then even if you don't give the live bacteria and you give the supernatant, you should see still the impact. And that is what's happening. So first of all, the heat kill is not working. So it's not something in the membrane of leptotrichia or a protein. A protein could be, but it's not something that it is from the cell membranes that is inducing the tumor. But if you add only the medium of the supernatant, is also working. So it is something extracellular produced by leptotrichia that is inducing the tumor progression. That was very, very interesting. Now, I switch a little bit. You try to understand also from the host what is happening. So we took the tumor from the mice that were injected with leptotrichia, and we compare it with the control. And we did many different things. So we look at the cytokine profiles, and you see many cytokines that have been connected with inflammation and the immune system to be increased in the presence of leptotrichia compared to the controls. And then we did also FACS analysis, so to see the immune cells, the immune cell populations, how they are distributed, how they are changing when you are going from when you are injecting this species. And we saw that the myeloid cell population is really expanding in the group that you have the injection with leptotrichia. And if you start zooming in, you find other subpopulations. You find that the M2 cell population is expanding. You see that also the GM DSC is also expanding. So this means that these cell populations have been linked with a more immunosuppressive environment. So you suppress the immune system with these changes. And even the T cell population that it is linked more to immune response positively, you see a decrease in this T cell population, both Th cells and T regulatory cells that decrease in. So in general, it appears that leptotrichia, it creates a more tumor-friendly environment. Now, have we done more? Yes, we have done more. So we have done single cell RNA sequencing in the tumors. And this is also very important because we can see specifically how the metabolism of the different immune cells, like the M2 cells that we observe here, is regulated in the presence of leptotrichia. So I think it's pretty interesting. But again, for the ones that are paying a lot of attention to what I say, I haven't really answered how leptotrichia is really promoting cancer. I think we have figured out what it does to the host, but how exactly is doing it, I haven't really told you yet. And I think I will not tell you. What I will tell you is that we have limited down to 6 metabolites that we are currently testing individual in cell lines. We have a pretty good idea. And what helped us a lot is something that you may think was a negative experiment. We tested 5 oral bacteria and only 1 worked. So you may think that you wasted a ton of money in order to do these mice experiments. But now we have a negative control, so we can see what does Leptotrichia produces that the other species cannot produce, because we have also the metabolomic analysis of the individual species. So it's really, I think for me, this research is really an amazing, like a game, like a puzzle that you try to figure out and put the pieces, and then you have this last piece that we are trying to put now and make the story complete. But I think it's already very exciting and a very expensive project. So if we come back to the original questions, how reliable is stool when you study colorectal cancer? I would say it is pretty reliable. You can get a lot of information from the stool, but definitely you cannot get the whole picture. And if you are trying to identify something new, probably the tissue is the way to go. What makes a microbe resident of the tumor tissue? I think it's obvious that the bacteria that they are present in the tumor tissue have a different metabolic potential. This metabolic potential could be linked with the progression of the cancer, but not always. It could be simply that they can tolerate the particular microenvironment of the tumor, and it's something that, as I said, in parallel, we also investigated I think in an interesting way. So we grew cancer cell lines, and we took the exometabolome and we were throwing it into this bacteria medium to see how they are going to respond. Does it inhibit their growth? Does it help their growth? So it would be nice to see also this. I don't have this result actually yet. It's in the analysis phase, but it could give some information why some bacteria that have nothing to do with the tumor progression are found there. Could be a competition that there is no space to go somewhere else. And which changes induced by gut bacteria that promote CRC? I think we have a pretty good idea of a few metabolites that are involved in M2 polarization, and this is what we are exploring even more. So with that, I think I'm still on time. I reached the end. If I had to put the complete list of people that they have been contributing in this project, you can imagine that there is really an endless list of people. I would like to only mention the most critical collaborators, external collaborators. Biljana Gigic from Heidelberg, she's heading the Colorectal Consortium, a fantastic cohort from in Germany. It's international, but she's heading the German part. So she played a major role with the clinical part. Clara Mello from Leibniz Institute for Aging in Vienna. She's doing a lot of work with the cancer cell lines. And of course, a very good collaborator and friend, Michael Zimmermann from EMBL for the work that he did with the metabolomics of the individual species. And with that, I have reached the end of my talk. I really hope in this always weird situation where you don't know if people are there or listening, and you're wondering if after 45 minutes you realize that you were offline. I really hope that everything went smoothly, and I'm happy to discuss with you things that were unclear. I will stop sharing now, but I can share again if you want to go back to a particular slide.
Thank you, Jen. Yes, everything went smooth. We could hear you and see this interesting talk.
Okay.
And we have also some question actually. I can read it out if you want in the Q&A section.
Yeah.
Okay. We have a question from Pau Perez. Biofilms are a known feature of a subset of CRC tumors, especially in right-sided CRC. Did you take this into consideration during your sample processing protocol?
I'm not sure if this was taken into account. I mean, we did for almost 6 months with an external collaborator the optimization of the extraction of the tissue microbiome. It really took a lot of time to select the right extraction buffers and protocols. Still, 85% of the reads that we generated were thrown away. It was host. They were not really thrown away because we could do some analysis for the host. If someone really wants to know the exact details of the protocol, please drop me an email and I'm happy to share it. Take it from the methods of the paper, because it will take some time until the paper is published, and send it to you. But it was really a lot of time and work for the optimization of that. And we needed to go, I think it was 20 or 25 giga per sample of sequencing in order to have enough bacterial reads. I don't know if the biofilm specifically was something that we took into consideration. It's a few years now that we did that, but I can share the protocol. I'm happy to do that.
Sure, we are happy to connect here on the platform. There's another question from Daisuke Suzuki. And a comment also, interesting talk. Yes, it was. I was fascinated by the tumor-resident bacteria which didn't appear in stool samples. Did you find any species which shown only enriched in the stool but not in tumor?
Yes. There is also the other way around, that there are stools that you can find significant, and that's why I'm also wondering how many of the results. But okay, let's put it like that. There are different mechanisms that the microbiome in your gut can impact the tumor progression. It could be something that is sitting on the tumor and producing something that really affects locally, but the gut bacteria that were found significantly different only in the stool doesn't mean that they don't have an impact, because these bacteria are maybe in even larger also quantities, and they have a systemic impact on the immune system. They are connected with metabolites, lipids that interact with the host, so they have a role. It's just different the roles and different the mechanisms that you are investigating when you are looking at the stool compared to when you are looking at the residence of the tumor. That's what I say. But you find different things, also different, unique, but many were in common. As I said, fusobacteria. Boom, you find them in every case.
Great, thanks. Another small question from Pau Perez. Were bacteria injected in the SC tumor or gavaged?
No, they were not gavased. It was
Gavaged.
directly in the tumor. Yes, they were
Okay.
not gavased.
Okay, great. One question
And there was a reason for that, is because these bacteria, we didn't find them to colonize really in the stool, so it were completely absent. So we thought that they will simply maybe host if we do oral gavage. So we decided let's put them in the tumor directly, so we increase the chances.
Great. Two questions I see from Fu only. What exactly was the input for the PCA, and how did you interpret these PCR plots? Can you say the stool is more variable than both the mucosa and the tumor, while tumor and mucosa are quite similar? And what are the nationalities and ethnicities in your patients?
Yes. So if I start with the last one, it's all from Germany. So I don't, at least are from German hospitals. So the majority should be Germans. But we didn't look into the ethnicity much in detail, because we had also a very small cohort, but it was German hospitals. Now for the PCs, if you mean the comparison that we did, this is I think a Procrustes analysis, where you try to find how similar are two matrices basically. The difference, so I see here the numbers, which is the sum of squares. And I see that it is 0.41, 0.43 when you compare stool with tumor and stool with mucosa, and 0.3 when you compare tumor with mucosa. But this is sum of squares. That means the lower the value, the strongest the correlation. So the strongest was the correlation between tumor and mucosa compared to stool with tumor or stool with mucosa.
Super. We have a lot of more questions, so I will go and we will see how
Yeah.
long we can do that. But it seems now at the end there's coming a lot of questions. From Joanna Kuto, is the leptotrachea conditioned medium also influencing the immune response?
That's a very good question. We didn't do that. We didn't do that. Uh, we focused on the live, so everything that we have done is with live. But another very smart people like you ask the following question: is it specific these immune responses to leptotrichia, or any gram-negative bacteria that you will add will cause the same effect? So that was a question that I heard in March. So we started again mice experiments, and we did it with 3 more bacteria, gram-negative bacteria. And I would say that only part of the things that I show you are very specific to leptotrichia. And now we are focusing on these very unique parts of the immune system that are only induced by leptotrichia. But the conditional medium, we didn't try it. Sorry, the medium we don't try it because we are trying to find out the individual metabolite, and we do it directly into the cell lines. So we throw the metabolite to the cell line. We don't do it in mice.
Okay, great. Yes, we have an interesting question from Kailesh Arda. Thanks for the great talk and fascinating study. Are you considering focusing on the metabolomics of different bacterial growth media, for example, CRC enriched versus control enriched, to identify functional properties?
Yes, exactly. So these 25 are guiding us. These 25 bacteria are guiding us, but the problem is that you don't really know which of these 25 are really good bacteria or bad bacteria, or they're simply sitting there. The experiment that I have not shown you is that we also did a screening of these 25 bacteria in cancer cell lines. So we have 7 cancer cell lines, and we used exometabolome. And by doing that, we try to see, because we couldn't do mice for 25 species. So we screen and see. Then from those, we are keeping the ones that they show some impact to the cancer cell line. And then we are looking to the metabolomic profile of those. Because if you look into all the 25, you don't know if they are having a causal relationship with the tumor, or, as I said, obviously some bacteria have no other space to sit, and they are going and sitting in the tumor, like the oral bacteria. Could be they are more used to the inflammatory environment, and they prefer to be there. But they are not doing anything to you bad, at least, as we see. So it's many things that you need to consider. But you are right that the metabolomic profile of the 25 are giving you already a hint where to look.
Great. Just thank you guys for this active discussion. So, Gianni, are you fine to stay a few minutes longer? Because we have some more questions here. Is it fine for you?
Yes, of course, yes.
Great. Then let's move forward. And if there's any question we can answer today, we will do it later, or you can reach out to Gianni.
Yeah.
We have a question from Joanna Kutu. Do you think the fungi are not so much studied because it's complicated to extract?
Yes, yes. It's many things. There are very low in abundance, so it's dominated, and we have not reached the point to know more about their functional potential. It's like that we were the limitations that we had when we were simply doing 16S. There are certain things that you can do, and then you stop because you don't know the functions. It's difficult. There is a very nice paper published in Cell, I think it was last year, where they isolated a huge number. It could be, maybe I'm mistaken, but I think it's something like 600 fungi from the human gut strains, and they sequence them. And now this is available. And using that, they claim that from the shotgun metagenomics, you can get also some functional information also for the fungi. We haven't tried it yet. We are setting it up to see. But I think with Novogene dropping the prices more and more always for shotgun metagenomics, if you go deeper in sequencing, plus these developments, maybe you can start getting additional information for the fungi. But it's very important. I think fungi, especially for the immune system, we know that they play a role, but it's more difficult to study and then how they do it because you don't get any more information. It's something they produce. It's something what they are doing. But I'm very much interested. I have it in all my projects, the ITS sequencing. Um, but yeah, it's true that, for example, for the tissue, it was impossible to get anything out from the metagenomic data. It was so low the number of reads that required other analysis, and it was not possible. But I will not discourage anyone from going with the fungi. I believe on that as well. It's just that, yeah, the focus was again bacteria.
Yeah, we have actually two questions from Andrea Proano Vasco. Very interesting talk. A diet rich in fat is a risk factor or CRC of CRC. Is leptotrichia associated with high fat diet? And the next one, do you know if there's a correlation between the abundance of leptotrichia and other cancers in the gastrointestinal tract?
Not other cancers in the gastrointestinal tract. We looked in general in tumors to see if is detected, and we found it. We found in other. I don't remember which cancers, but because we're afraid whenever you come with something so new, you're afraid that. Someone may say that you were spitting on the tumor, and that's why you find leptotrichia. So we looked into other tumors, and we could detect leptotrichia, but I don't know if it has anything to do with high fat. I'm sorry, I have no idea, and we didn't look into that. And it's difficult to look into that because it's not detected in the stool. In stool, I have many cohorts, very large cohorts with dietary information, so I could make this connection. But you will need a cohort that has tissue metagenomics and dietary information, and I'm not aware of any. Yeah.
Okay, thanks. We have more from Victor Haas. Thanks for the talk. Do you have an hypothesis on how the bacteria translocate from the oral cavity to the tumor? And do you know if altered oral microbiota could play a role here?
The second question was which oral what was
Do
it?
you know if altered oral microbiota could play a role here? So do you have an hypothesis on how bacteria translocate from oral to tumor?
Mm.
And do you know if changes in the oral microbiota could play a role here?
There are different theories about this. One theory says that in reality, it's not that you have more oral bacteria under specific conditions, as in my case, the colorectal cancer, but you have a dysbiotic microbiome. So you have very low diversity, and that allows you to capture also the presence of oral bacteria. That in reality is not that you have more. It's the same, but it's that you have the opportunity to detect them with the sequencing. That's one. I cannot say that it's one or the other. It's, as you said, it's hypothesis. It's not something that I'm looking how they ended up there. But for me, there is a huge competition in the gut for resources. It's not easy to get some space down there. Otherwise, we wouldn't have this problem that we give antibiotics and 2 days later you don't find anything, right? I mean, sorry, probiotics. I don't know if I said probiotics or antibiotics. That probiotics, we know now that they cannot stay, they cannot colonize. So it's very competitive environment. And then suddenly you have a niche where probably the conditions are not ideal for the gut bacteria, but could be ideal for another set of bacteria. With the saliva, we constantly bring oral bacteria down. So it could be a combination of low diversity. These people have been treated, maybe they have therapies before, chemotherapy, or some of them, we have the antibiotics profiles. They have taken antibiotics the last 6 months or not, different medications. Maybe they have created a dysbiotic environment, low diversity, and then they can these oral bacteria can find a niche. But that's a hypothesis, yeah. I believe it's easy to test it. I mean, if you want to test it, it's easy. You can start adding these oral bacteria under a different perturbation conditions. One of your in the audience said high fat. You try different diets, you try different antibiotics, and then you find out when do these oral bacteria manage to get a hold in the gut. So I think it's also an interesting idea for a paper.
Sure, sure. I guess we will do one last question. I think otherwise it would take into the afternoon here. But yeah, there are some more questions. But as the last one for today's webinar, from David Cisneros, great talk. For species that are enriched in the tumor microenvironment but do not promote CRC, do you consider looking at bacteria interactions, e.g., like for example, if Leptotrechia cooperatively promotes growth of the other species?
Very good point. Yes, that's a very interesting approach to see whether Leptotrechia also needs the others that are present there to support. That's very. I don't plan to do it because I need to finish. You see how much analysis has gone into this, but it's an excellent suggestion to make pairwise co-cultures of Leptotrechia and find which are the supporters, because this can have also therapeutic, open therapeutic possibilities, right? Maybe you cannot hit Leptotrechia directly, but if you can disturb the network that supports Leptotrechia, the way that I could do it, and maybe I can add it since we are still writing the paper, is I can do it in silico. So I can use genome scale metabolic models of all the different bacteria and study in silico whether Leptotrechia is increasing the growth in the presence of the exometabolome of the others. So it's a good idea. I can do it in silico. I hadn't considered it, but I can do it and even keep it back for reviewers like you that may ask additional things. Yeah.
Okay, great. I think we will stop at this point, but we can come back to you for the other
Yeah.
questions. Or you can connect to us and Gianni, of course, as this platform is there as serving a platform to connect you. So thanks, Gianni, for this. It's always a
Thank
pleasure
you very much. Thank
to
you.
listening to
I
you.
really mean it. Thank you very much, because all these questions are things that you need to consider when you are writing the story. It was really excellent feedback for us. Thank you very much.
Great. So yeah, thanks again, and also thank for everyone joining today. Yeah, stay connected to us, reach out to us or to Gianni. for any collaboration or question you have. And I guess seeing that active discussion, I think we will, of course, continue offering that platform for you guys. Yes, thanks for being part, and see you hopefully at future events, and enjoy the rest of your day.
Thank you very much. Okay, have a nice
Okay,
day. Bye bye. Thank you, Adrian.
nice day. Bye bye.